This is the second half of Unit 1 in Micro 240, and we're going to start out talking about hemoflagellates, and these are parasites that have flagella and they're found in the blood. The two organisms that you need to know about are Leishmania and Trypanosoma. And there's some terms that you need to know. In Leishmania, these are the phases, the amastigote and the promastigote. The amastigote is a small oval organism that's seen in the blood smear. It doesn't have any flagella, and it can be intracellular. It has a kinetoplast, and this is the kinetoplast, and this is the nucleus in the organism. It also has a blepharoplast, which is just a structure. It is where its axoneme arises, and we talked about that word in the first half. The promastigote is found in the vector, so we don't see it in the patient sample, and it has flagella, and this is a diagram of what that looks like. In Trypanosoma, there's two phases. The trypamastigote is seen in the human, and the epimastigote is seen in the vector, and both of these are they have flagella. They both have an undulating membrane, a nucleus, a kinetoplast. So you need to know these two are found in Trypanosoma, and the amastigote and promastigote are in Leishmania. And that's a little bit repetitive, but you can look at that. Okay, Leishmania brasiliensis. And let me just say this, I have tried to put on these PowerPoint slides the things that are unique to each organism to better help you study. However, I don't want you to study the PowerPoints on their own. They will definitely help you a lot, but there could be some information that is not included on the PowerPoints that's in your lecture notes that'll be on your test. So the first one is found in South America in rainforests where chicle sap is used for chewing gum. If you want to know more about that, just Google it. The vector is a sandfly, so the organism is in the sandfly and it bites a human and transmits the promastigotes in the human. It goes to the RES system, and then it transforms into the amastigote. The amastigote is the diagnostic phase. This is the little oval-looking organism with the nucleus and the kinetoplast. This is in a peripheral smear, but this over here is a skin lesion, and this is a white cell. And the amastigotes are inside this white cell. Okay, it causes mucocutaneous leishmaniasis or aspundia, and these patients have ulcers, and it destroys the cartilage, and it causes deformities of it's usually on the face. The patients have secondary bacterial infections, and the skin areas can be biopsied to look for the organism. So it's either a Wright or a Giemsa stain, and the organism is 2 to 5 micrometers, and it has a nucleus and a kinetoplast, and this is what they look like. Notice at the top, we're going to talk about 3 different species of Leishmania, and they all look the same. So when you're in lab or when you're at the hospital working, if you see these, then you're supposed to report them out as just Leishmania species. You are not going to be able to tell the difference between the species by looking on the slide. The physician should be able to tell what organism or what species the patient has by their signs and symptoms, where they're infected at, and what country they've been to. This is another picture of probably a skin biopsy, and these are the organisms inside the white cell. Okay, Leishmania donovani complex. This is the most severe disease. There's 3 different members in this group, and the correct titles of these are Leishmania donovani chagassi, and Leishmania donovani and Leishmania donovani infantum. You're supposed to, it has all 3 names. So you need to know the vectors. In Chagassi, it's the Leusdomia sandfly. Donovani in infantum is the phlebotomist sandfly. A gem sustain should be done, and this is done on blood, bone marrow, or the lymph nodes. And also a buffy coat can be performed of blood from the patient. It looks just like the other organism. The patients have visceral leishmaniasis, Kala-Azar, dum-dum, or black fever. And these are all the typical, they're all the same disease. The patients have a characteristic darkening of the skin, and these patients could die. It can damage a lot of the organs in the body and eventually kill them. Okay, Leishmania tropica. It has the same life cycle as the other 2. It causes bulls on the skin, usually the face or the extremities, and a biopsy of those can be done from their sore. And the diseases are called cutaneous leishmaniasis, old world leishmaniasis, oriental sore, Baghdad bull, or Delhi bull. And again, it looks just like the other ones. Okay, Trypanosoma brucei-gambinese. There are 3 different species of Trypanosoma that you're going to study, and just like leishmaniasis, you can't tell the difference between the species on the slide. So you would just report out Trypanosoma species. Okay, with the first one, it causes a sleeping sickness from tsetse flies, and the specimens are blood, lymph nodes, and spinal fluid, depending on how bad the patient is. It causes West African sleeping sickness. You need to know that. The patients will have ulcers where they got bitten by the fly. They have something called, or they may have something called Winterbottom sign, which is just where their lymph node it has enlarged, and you can see a picture of that. The organism can be spread in severe cases and get into their spinal fluid and cause retardation and meningoencephalitis and even death. Okay, the next one is Brucei rhodesiensis, and this one is the more virulent organism and can cause a worse infection. This one is the cause of East African sleeping sickness, and this disease is usually more rapid and acute. The patients get sicker faster. It has the same diagnosis. You do a slide, and it can spread to the other organs. The patients don't have the enlarged lymph nodes, though. Okay, this is the Trypanosoma trypomastigotes, and this is what they look like, especially Trypanosoma cruzi. We hadn't got to that one, but cruzi will typically look C and U shaped. but still they're so similar that you just report them out as Trypanosoma species. So they're 15 to 20 micrometers. They're easy to spot even on the 40x objective. They have a large nucleus, which is right here. The kinetoplast is right here. And this is their undulating membrane right here. Okay, this is Cruzi. It's seen in South America. It's transmitted by the Redidavid bug, the kissing bug, and Cononos bug. And these are usually found in open designs of homes. And the fly will defecate on the patient, and it's scratched into their skin. The organism is scratched into the patient's skin. It's usually seen in children more often. A Giemsa or a Wright stain can be used to find this organism. It causes Chagas disease, and the first symptom is the erythematous nodule that's usually on the face from where the infection happened. There's also something called Ramona's sign, which is edema around the eyes, which is what's wrong with this baby's eye. The acute disease could cause death. They have fever and chills. The chronic disease, they have especially the heart and the skeletal muscle are affected, but could also be the colon. And it's usually seen in children under 5. Okay, the Leishmania and Trypanosoma is found in chapter 28 in your book, and it starts on page 645 and 646. It's a good section. It's where my notes came from. Okay, the next thing that we're going to talk about is the malaria parasites, and they're in that same chapter 28, and it starts on page 650. Malaria are classified as sporozoa, and their scientific name is Plasmodium, and there's 4 that you've got to know. They're considered obligate intracellular parasites, and what this means is they have to have a cell to live. They invade the cell, and they need the cell to survive. Okay, these are the different forms, and you need to know what these are for lab so you know what you're looking at. So this is the first phase that we see in patients. This is like the baby phase. And they're called ring form trophs. So they have a little ring, and they have a small purplish dot, which makes it supposed to look like a ring on your finger. So this would be like the diamond, and then there's a ring. So the ring is the cytoplasm, and the dot is chromatin. And then this starts to grow and fill in, and it's called a developing troph. So it will start to take up more space in the red cell. There's more cytoplasm. It starts to get thicker. Then it turns into a schizont. The schizont will be filled, when it gets mature, it will be filled with these small, these right here, of chromatin mass, and they're called merozoites. And one of the ways that we identify which organism it is by how many of these merozoites are in the parasite. And then they turn into gametocytes. There's a microgametocyte, which is the male, and a macrogametocyte, which is the female. They almost fill up the entire red cell. You can't even see where the red cell was, but the chromatin is usually pink to purple, and it can be darker, and it doesn't have much empty space in it where you can see the red cell. Okay, With the ring form, we've already talked about this. This is the little ring form with the chromatin dot. This is an early troph. It gets in the red cell. The developing troph starts to take up more space. The ring gets thicker. This is the schizont with the merozoites, and this is a type of asexual sporozoa, asexual replication. And then the gametocytes are sexual, and these are just two different types of gametocytes. Okay, in the lab, whenever we have a patient who has like consistent fever, it's important to know their history and their travel history, because in places where malaria is prevalent, then the doctor will suspect it could be that, and what's done is just an EDTA sample, and it's important to collect between the paroxysms, which is the period between chills and fevers. The chills are supposed to be in response to the schizont developing, and the fever is supposed to be in response to the merozoites being released. Blood should be collected every 6 to 12 hours up to 48 hours to make sure that the patient is negative. So if you get a positive on the first slide, you can stop. Usually there's 2 slides that are made, a thick one in which you just put a big old drop and you don't smear it out. This is to look to see if there's any organisms or not. So you're just looking to see if you see anything suspicious. If you do, then you use the thin film that's spread out with a feathered edge, and you use the thin film to identify the organisms. Some people do a procedure that will lyse the hemoglobin over here in the thick one so that you can see it better. It really doesn't look this thick if you lyse the hemoglobin. Then the organisms are kind of out there for you to see them easier. The GEMSA stain is preferred, but also the right stain is used. It's important to know where the patient's been and the red cell morphology. There's also some faster tests that have developed that use DNA probes and the ELISA method to identify. Okay, the malaria life cycle. This diagram came out of your book, but there's a lot of stuff over here that you don't need to know, like what happens in the fly. It's these ooconites you don't need to know, this oocyst. So this is I just thought this was a good diagram for the part you do need to know. It's on page 652 if you want to look at it closer. But the vector of this organism is the Anopheles mosquito, and the mosquito bites the patient and they inject these sporozoites. And the process of them growing and multiplying occurs, and this is called schizogony. So schizogony occurs. They will travel to the parenchymal cells of the liver, and they mature into ring-formed trophs, and then they're released and they invade red cells. And in the red cells, they feed off of the hemoglobin. So the gametocyte is eaten by the mosquito. In the sexual phase is found in the vector. The asexual phase is found in the human. And if you go back to this chart, you can see this is in the mosquito, and the gametocytes will mate, and they form this this parasite over here. Whenever the mosquito bites a human, then the sporozoites are released into the patient's system. Schizogony occurs and the merozoites are released. They invade the red cells, and then this is the maturation process of inside the bloodstream. Okay, the malaria has been transmitted by blood transfusion, and this is one of the reasons why the Red Cross asks a bunch of questions about where you've traveled. It's also been transmitted by needles and from mother to baby. The patient has intermittent chills, and they can last up to 20 hours. Some of these infections can reoccur. The main symptoms are kind of similar to the flu: fever, chills, fatigue, and headache. They can have nausea and vomiting. They can have damage to their liver and their spleen. Treatment is based on which organism the patient has. Okay, now we're going to start talking about this, and I hope this doesn't blow your mind any more than it's already blown. We're going to talk about these 4 different organisms, and you need to be able to tell the difference between them on the slides. So there is a really good comparison chart on page 653 that has can tell you the difference between them. Okay, the Plasmodium vivax has a 48-hour life cycle. You need to know these numbers of their life cycles. Vivax infects young red cells, so one of the things that you do to help you identify them is to look at the size and the morphology of the red cells. So it infects young red cells, so these are cells that would probably be polychromatic, and they're going to be 1 to 1/2 to 2 times the normal size of a red cell, so they're going to be enlarged. Okay, the ring form takes up about 1/3 the diameter of the red cell, and it has 1 chromatin dot and a vacuole. The developing troph is usually irregular, amoeboid. It can have these things called Schaffner's dots, and these are just condensed hemoglobin. It can have large vacuoles. In the schizont, it's supposed to have 12 to 24 merozoites and some golden brown pigment. In the blood smear, you typically will see a wide range of stages. So you will see schizonts, developing trophs, ring forms, gametocytes. These patients typically have photophobia, which is sensitivity to light. You need to know that. That's the only one that does that. Okay, this is the ring form. This one has a large chromatin dot and a vacuole, just like we said it's supposed to. The vivax developing troph here is a regular and amoeboid. See how it looks like it has a pseudopod? It looks amoeboid. This is the vivax schizont, and if you count these, there should be 12 to 24. They typically have this golden brown pigment. And these are the gametocytes. There's not a whole lot that you can tell between the organisms about these round gametocytes. They're typically pink to purple with brown pigment in them. And this is what Schaffner's dots look like. They look kind of like stippling all over the red cell. Okay, Plasmodium ovale has a 48-hour life cycle also. It infects young red cells. One thing about ovale is it can destroy the or distort the red cell. Do you see how around these red cells it looks a little ragged? That's what it will do to them. And this is called, the red cell is said to be fimbriated when it looks like this. And ovale is the only one that can cause fimbriation. The ring is 1/3 of the diameter of the red cell. It's the ring is larger than P. vivax, it can have up to 2 chromatin dots instead of just 1. And my pictures just have 1, but it can have up to 2. Fimbriation, it can have Schaffner's dots. In the Schizont, it can have 6 to 12, which is a lot less than Vivax. Vivax has 12 to 24. And Ovale averages 8. The Schaffner's forms can be found in all stages. This is least common of all of them, and it's confined to Africa. This is the Schizont, so let's try to count the merozoites. 1, 2, 3, 4, 5, 6, 7, 8, and if that's 1, 9, I'm not sure. So that fits, and this even looks like it has a little bit of fimbriae coming off of it. This is a gametocyte. They look pink to purple, little bit of brown pigment, and the outside of the red cell is distorted a little bit. Okay, with malariae, this one has a 72-hour life cycle, and the previous two only had 48. This one also infects mature red cells, so the red cells should look like the normal size. The ring forms are a lot smaller, and they will look like they have a heavy chromatin dot, and the vacuole in the center may look like it's filled in. And this is what I mean. This is smaller compared to the entire red cell. The chromatin dot is heavier, and it looks like there's some stuff inside of the ring. The troph can be a band or a bar shape like this. The Schizont can have 6 to 12. This is just like the previous one. But if you see something that's supposed to look like a daisy petal, then you know it's malariae. They're kind of arranged to look, I mean, I know it doesn't really look like a petal, but they're not arranged like the other ones. The Schizonts usually fill all of the red cells, and it has something called Siemens dots. The Siemens dots are cytoplasm, and that's what that is. And this over here is just an artifact. Okay, Falciparum has 36 to 48 hour life cycle, and it's the organism that has the highest parasitemia. So this means that there's going to be a lot more red cells infected on the slide. It's the most severe and the most often to be fatal. Sometimes only the ring forms will be seen on the blood smear. So this is something that you need to remember. The ring is circular. It has 1 chromatin dot, or sometimes they look like a headphone, like this one does, and this one. 1/5 of the red cell is taken up. It has delicate rings, so they're not, the cytoplasm is not thick like these two are. These do look a little thick, but these are more delicate and thin. Multiple rings are common, and they have something called, or they do something called appliques. The applique refers to it looking like it's trying to exit the red cell. The trophs have heavy rings. The Schizonts are only seen in severe infections. They have 8 to 36 merozoites, an average of 20. It fills 2/3 of the cell. This is another thing that is unique to Falciparum. It has banana, it refers to them as sausage, crescent, or banana-shaped gametocytes. And if you see any that look like this, it's falciparum. None of the rest of them will have that banana shape. It also has Marr's dots, which are made up of protein, and they're dark. This is a picture. Dark little spots. They can be wedge-shaped, and it's hard to see unless the stain is really, really good. Okay, I think the bottom of my slide is cut off right here, but you do need to know for sure that falciparum causes black water fever, in which the patient has a sudden massive intravascular hemolysis, and they have hemoglobinuria. They have tubular necrosis, which is referring to the tubules in the kidneys, acute renal failure, and nephrotic syndrome, which refers to the kidneys as well. It can even cause death. Okay, the next organisms are also sporozoa, and it's called Babesia microti. And Babesia looks a lot like Plasmodium because they have these ring forms. But how you tell the difference is that Babesia will have rings on the inside and the outside of the red cells. And there won't be anything that looks like schizonts or developing trophs or gametocytes. There won't be any of this brown pigment that you see with malaria. Okay, so they look like the ring forms. They're inside and outside the red cells. They have 2 to 6. They're 2 to 6 micrometers, and they have small chromatin dots, no pigment. Some of them will have a cross or like this that looks like a Maltese cross. It's usually found in rodents. It's a parasite of rodents. The vector is called an Ixodes tick. It can cause hemolytic anemia. It can be transmitted from mother to baby and through blood transfusion. The patients have a lot of the similar symptoms as Plasmodium, and again, it's done. The diagnosis is done through Giemsa or Wright stain, and this is on page 656. Okay, we're almost getting to the end of this unit. Toxoplasma gondii. This one is a little bit different. It has sporozoites also, and it forms, it has an encysted form of its zygote. And in those, it has mature sporozoites. It's round to oval in shape, and it's transparent. This is what it looks like. These are the tachyzoites. These are the actively multiplying crescent-shaped part of the organism. It has a centrally located nucleus, and these are motile. The bradiozoites look like these. They're very similar. They are smaller. They're found in clusters, and they can make a cyst. And in the cyst, there may be 50 to 1,000 bradiozoites inside of it. Okay, with Toxoplasma, the AIDS patients are very susceptible. Toxoplasmosis looks a lot like mono. And one of the things that you need to remember about this organism is it's dangerous for babies. So therefore, the mother, when you find out you're pregnant, the doctor will order an antibody level for Toxoplasma to make sure you have antibodies to it. If you don't, then it could infect the baby and cause a lot of problems. Over 50% of the United States has been exposed and has antibodies. So you don't even know you were exposed. It's not going to make us sick. The definitive host is the cats, and the cats eat the rats that have cysts in them. Whenever a pregnant woman is carrying her baby, she's supposed to stay away from cat litter boxes. And again, it's found in AIDS patients, and who is most at risk are AIDS patients and unborn babies. Okay, Isospora belli is oval and transparent. It has several stages. The sporoblast will divide into two sporoblasts and it becomes a sporocyst. The sporocyst has 4 sausage-shaped sporozoites that are surrounded by a 2-layered cell wall. It's increased in AIDS patients. We're back to stool again. This infects the GI tract and the patient has diarrhea. It can cause eosinophilia and Charcot-Leyden crystals can be seen. It also causes malabsorption and increased fecal fat. The fat from your food should be reabsorbed by the intestines. And in this, it causes problems with absorption, so there's more fat seen in the stool. And also, notice this is a modified acid-fast stain. This is used to identify Isospora and Cryptosporidium. So the organism will be hot pink. And this is the 2 sporozoites in the organism. Okay, Cryptosporidium parvum, this is what it looks like, and again, this is an acid-fast stain. The organism is 4 to 6 micrometers. It has 4 small sporozoites, 1, 2, 3, 4. It doesn't say this in your notes, but one thing to notice is when it stains, it has a slight halo around the organism where the stain isn't taken up, and right here. It has a thick wall, 1 to 6 granules can be seen. It can look like yeast or white cells or red cells on these slides. It's often seen in daycare centers, travelers, and immunocompromised. The iodine is the best method. A lot of people still do modified acid-fast stains, however. Patients have cryptosporidiosis, which causes diarrhea, and it can be a severe diarrhea or diarrhea that's not a big deal, and it can be self-limited. This organism will alter the pressure in the intestines, causing a cholera-like diarrhea in which they lose a lot of fluid. They can lose from 3 to 6 liters a day. It's usually found in contaminated water, and if you want to pull the PowerPoint up and click on that, this is Cryptosporidium that was in the news recently in the summer 2016, and it was found in a swimming pool, and I think it was more than one swimming pool in the country that was closed because of that. Okay, Balantidium coli. It's the largest parasitic protozoan in this group, and why don't I have a PowerPoint for that? Who knows? I'm sorry. This is what you need to know for this one. It's the largest parasitic protozoan. It's classified as a ciliate because it has cilia, and these are the cilia around the organism. It's the only human pathogen in its group. Again, it's very large. It has 2 nuclei, one kidney bean shaped macronuclei and one small round nuclei. It has visible contractile vacuoles for food, a cytostome, and again it has cilia around it. The cyst is double-walled. This looks double-walled also, but it has cilia in between the layers. This is the kidney bean macronucleus, and this is the small cytostome. The pig is the known reservoir. The human is an accidental host. Patients can lose a lot of fluid from diarrhea. It usually comes from contaminated food or water, and it can spread to other organs and also cause anemia. This picture is actually the next organism, Blastocystis hominis. This organism has a huge vacuole in the center right here, and it has 2 nuclei, one over here and one over here, and it can have 2 to 4 nuclei, and the vacuole consumes about 90% of the organism. This is the huge vacuole here that's stained. This is the vacuole. It's found worldwide. It also causes diarrhea. This organism is used to be classified as a yeast, and now it's in a parasite, in the parasite families. What you need to know about this is you use iodine wet preps. If you do something that uses saline and water, the organism will lyse and there will be false negative results. You won't be able to see the organism. And I don't have a PowerPoint for that either. I don't know what happened to these PowerPoints. I'm sorry. Okay, the next one is Pneumocystis jirovecii. I'm not sure how to pronounce that. It used to be Pneumocystis carinii, and they recently changed the name. These are easy to identify also, in my opinion. This is what they look like here. The trophs are 2 to 4, ovoid or amoeboid. The cysts are roundish with thick walls. It has a nucleus 4 to 8, unorganized or in a rosette. This is most often seen in AIDS patients. It infects the lungs. The patient can inhale these cysts and get in the lungs, and they rupture. The specimens are usually sputum, lung biopsy, bronchial washings or brushings. The Gamori silver stain is the best method, and this is the leading cause of death in AIDS patients. When they get infected with this, it causes a pneumonia called atypical interstitial plasma cell pneumonia, and that is the leading cause of death in AIDS patients. It also is related to a malignant skin disease called Kaposi's sarcoma.